Morning all. Today topic is biosynthesis of purine nucleotide. So many compounds contribute to the purine ring of the nucleotide. Many compounds contribute to the purine ring of nucleotide. Here N1. So nitrogen 1 of purine. This is the purine ring in which N1 is derived from the amino group of aspartate. It is comes under amino group. It is derived from amino group of aspartate. Okay, and C2 and C8 is arises from formate of N10 pharmyl THF tetrahydrofolate. C2C8 is derived from N10 pharmyl tetrahydrofolate. Okay, and N3 and N9 is obtained from amide group of glutamine. N3 and N9 is arises from or obtained from glutamine. It is amide group. Okay, and C4 and C5 and C7, these three C4 C5 C3 C5 and N7, these three are contributed by glycine. And C6 is directly comes from carbon dioxide. C6 is directly comes from carbon dioxide. Okay, so it should be remember that the purine bases are not synthesized as such, but they are formed as ribonucleotides. Okay, let me discuss about pathway. So biosynthesis, in which D-ribose 5-phosphate, nothing but ribose 5-phosphate is derived from a HMP shunt of carbohydrate. Okay, in the HMP shunt, the end product is ribose 5-phosphate. It is derived from HMP shunt of carbohydrate. The ribose 5-phosphate is involved in biosynthesis of purine, or the starting material of purine nucleotide. For that, we have to discuss one point. Liver is the major site for purine nucleotide synthesis. Erythrocytes, RBC, and polymorphonuclear leukocytes and brain cannot produce purine. Only liver can synthesize or produces purine nucleotide. Okay, so the starting material of purine nucleotide biosynthesis is ribose 5-phosphate. Here the ribose 5-phosphate react with ATP and produces 5-phosphoribosyl pyrophosphate, PRPP. Okay, so here the ribose 5-phosphate is meet with or react with ATP in the presence of phosphoribosyl pyrophosphate synthase and produces 5-phosphoribosyl pyrophosphate. So here in the second reaction, glutamine. So glutamine transfers its amide nitrogen to PRPP. It transfer their amide nitrogen group to phosphoribosyl pyrophosphate and produces phosphoribosyl amine So phosphoribosyl combined with glycine in the presence of ATP and produces glycinamide ribosyl 5-phosphate. So here next, N5-N10 methyl tetrahydrofolate, it will donates the pharmyl group and produces pharmyl glycinamide ribosyl 5-phosphate. Glycinamide ribosyl 5-phosphate, it will be formed by 5-phosphoribosyl amine. So here the N5-N10 methyl tetrahydrofolate transfers or donates pharmyl group and produces pharmyl glycinamide ribosyl 5 phosphate. So next, glutamine donates the second amide nitrogen group and form pharmyl glycinamide ribosyl 5 phosphate in the presence of synthetase. So pharmyl glycinamide ribosyl 5 phosphate react with ATP and forms 5 amino imidazole ribosyl 5 phosphate. Okay, here the imidazole ring of purine is closed in the ATP dependent reaction and to yield 5 amino imidazole ribosyl 5 phosphate. Okay, after that carboxylation, due to the carboxylation it will finally forms 5 amino imidazole carboxylate ribosyl 5 phosphate. So here aspartate condenses with 5 amino imidazole carboxylate ribose 5 phosphate and finally form 5 amino imidazole 4 succinyl carboxamide ribosyl 5 phosphate. Here it will, in this adenosuccinate lyase cleavage the fumarate succinyl, it is comes in citric acid cycle succinyl CoA fumarate malate oxaloacetic acid. So in which the succinyl converted into fumarate, so this fumarate is cleavaged by adenosuccinate lyase and finally it will form 5 amino imidazole 4 carboxamide ribosyl. And here N10 pharmyl tetrahydrofolate donates 1 carbon and form 5 pharm amino imidazole 4 carboxylamide ribosyl 5 phosphate. The final reaction is catalyzed by cyclohydrolase and finally form inosine monophosphate, nothing but IMP. Okay, so this is all about purine nucleotide metabolism. So నోట్స్ ఇది క్లియర్గా ఒకసారి చూసుకొని ఫోటో అయినా తీసుకోవచ్చండి. బేసికల్లీ ఇట్ ఈస్ ఫర్ తెలుగు మీడియం స్టూడెంట్స్.